so fortunate to have ARCS members able to visit with you. Let's use these minutes of your precious time that we have to hear what's happening with you and with your
Yes.
research and what you plan to be doing. So I do want to begin by making sure that everybody realizes that Leslie Petter is our chapter president, the ARCS Atlanta chapter president, and Caroline Harden is the chair of our Fund Development Committee. And I'll just mention on behalf of Northside that Northside has sponsored 5 ARCS scholars since it joined the ARCS family. 2 of them have graduated, and you are the other 3. And we're delighted to have Northside as a sponsor and to have you as ARCS scholars. So let's start off with the 3rd-year scholar in the group, and that is Christian. And you have done some amazing things since you have been at the University of Georgia. But tell us about your latest achievements and tell everybody what kind of research you're doing.
Well, I'll start off by, of course, thanking all the ARCS funding. And I'll just say, last year I was fortunate enough to be selected, or not last year, sorry, the year before. 2020 just kind of got skipped over. I forget about that.
Yeah.
But in 2019, I was able to attend a very, prestigious, because they only take 20 scientists, and graduate students are included in that. So it's, young professors, postdocs, and some graduate students. And I was able to go as a graduate student to go spend 6 weeks to learn basically the premier techniques in reproductive biology. So one of the cool things we got to do was we got to use a gene editing technology to make glow-in-the-dark mice. And then we performed IVF to implant the mouse embryos into a mouse. And then she gave birth to baby mice that could glow in the dark.
Oh, gosh.
And so
Goodness.
that technique would be more applicable to be, say, in the future, if someone has a genetic condition, we might be able to correct some of those things and then still have a live birth later in life. But in research, those techniques are very useful to study how certain genes affect reproductive biology. So ARCS basically provided all the funding for me to go do that course, which is, if you can imagine, to spend 6 weeks somewhere, it costs a lot of money. So ARCS funded pretty much that whole thing. So that was kind of the big thing that I was so, thankful for that ARCS allowed me to do. So I just wanted to get that off before I kind of get into some more of my research project stuff, just because that's a cool story. All right, but I will let y'all know, as far as research wise, we published a paper in 2019 that showed that we were the first lab to ever show this, that a single human relevant dose of a common chemotherapeutic drug altered the mouse uterine response to estrogen. So basically what we showed was that we treated these mice with chemo drugs, and then we challenged them with common hormones that are found throughout the female body later in life and saw how the uterus responds to that. And so estrogen is a key hormone involved in early pregnancy. So you can see how we've showed that these chemotherapeutic drugs can cause long-term changes to how the uterus responds to hormones that are important to pregnancy. And so, each year, tens of thousands of young women are exposed to these chemotherapeutic drugs. Doctors aren't really required to discuss infertility or any kind of fertility preservation methods. So what we're trying to do is explore if there are any functional changes. Do some of those changes that we saw in the uterus actually lead to infertility or lead to complications in pregnancy later in life? And so right now we've got a big project going that involves performing surgery on mice, which I'm actually scheduled to do at 12. So I'm hoping we can get out of here at noon. But again, ARCS, the funding that I used to attend that 6-week program led me to learn these surgical techniques that I'm going to use today. So, I'm very thankful for that. And I hope this shows how applicable this funding can be. I mean, of course, some of it went to my rent so I could live in downtown Athens and have fun. But, you know, a lot of it went to trying to learn new techniques for my science. So I'm very thankful. And I don't know if you're going to do questions now or if you kind of want to let everyone go and do questions, but I'm happy to answer.
Let's circle through and hear what everybody is working on, and try to keep it to just a couple of minutes, just very high level. But Katie, will you introduce yourself and tell us what you're doing at Georgia Tech?
Sure. Hi, everyone. I'm Katie Young. I am a 5th year BME PhD student, actually in the joint program between Georgia Tech and Emory. And so I've spent the past 5 years working on studying cancer cell mechanics. There's this wild thing where cancer cells are softer than their normal counterparts. And in fact, metastatic cells, the ones that spread throughout the body and are the real danger with cancer, those are even softer than their other cancerous counterparts. So I have come up with a method for studying those mechanics on the single cell level, and at the same time collect gene expression information for those same cells, so that we could hopefully better understand this metastatic process and this metastatic cell softening process, with the hope of figuring out how we could potentially stop that. And yes, so I'm so thankful for the ARCS funding that has allowed me to work on this research, and excited to get to hear from other scholars and talk more with y'all as well.
That's great. Jessica, thank you for being here.
Yes.
wish we could see your face, but tell us about yourself.
Yes, I'm sorry for the mask, but
No
I am I'm Jess Hoffman. I'm a 3rd year grad student at Emory, and I'm in the molecular and systems pharmacology program. But my research is interested in cell therapy for children with congenital heart disease. And so we're working on building predictive models to predict patient outcomes for these cell therapies. So in my most recent project, which we're hopefully submitting for publication soon, we are working with a phase 1 clinical trial, which uses donor-derived bone marrow stem cells. And so these stem cells were placed in the hearts of children with a very severe form of congenital heart disease. And so what my work tries to do is figure out if we can predict whether or not it's going to have the most beneficial outcomes. So try and select for optimal donors for stem cells. I was a math major in undergraduate, so I love bridging my 2 worlds of math and bio. And so I run lots of RNA sequencing experiments, collect gigabytes and gigabytes of data, and then I try to build predictive models with different machine learning algorithms.
Wow.
And so those are my research interests. And thank you, ARCS. This year, I've spent the money converting my apartment into an at-home place that I can do research, buying a computer that I can run these analyses on, because we were limited in laboratory time, obviously, due to the COVID-19 pandemic. And so this has just been a blessing. So thank you.
That's great. Well, do you scholars have any questions for ARCS Foundation? And then I'll also ask Leslie and Caroline to, I'll give them a moment to ask some questions of you. But do you scholars have a question or for one of us?
Did it be that a potential ARCS scholar might be the first woman on the moon?
Oh, I
Yeah.
know, Jessica.
Yeah,
Is-
Jessica Meir, that the Artemis program, you read that.
Yeah, I
Yes.
thought that was crazy. I mean, that's awesome.
Isn't that amazing?
Did you see the film she did from out on the space station? She's
No.
floating around and her hair's going all over the place. And she thanked ARCS. She gave a big shout out to ARCS.
From the space station?
Yes.
Yeah,
From
it's
the
on,
space
amazing.
station.
it's on the homepage of the ARCS National website. So if you go to arcsfoundation.org, it's just a minute long, minute and a half long video, but she speaks directly to the ARCS members and thanks us for the funding that we gave her when she was in at university, and talks about the importance of our mission. And we just all loved that.
Yeah,
So,
that was awesome.
It's
Well-
so great for little children to see somebody on the space station.
I know.
Almost as fun as it was when, in our day, ladies, when we watched the first person walk up, like
Wow.
get off the,
Right,
walk on the moon. That was so cool.
it was.
But I loved it.
So I want to ask the scholars, is there anything in your past that prompted you to do the kind of research that you're doing now? I mean, Christians working on women's health, I mean, that seems to be a strange, transition there. And Katie's working on cancer in the cancer area, some things that will impact cancer treatment and diagnosis in the future. And Jessica's already alluded to her math background, but is there anything that drew you to this particular research that you're doing?
I can start. I don't think it's probably very surprising to anyone with cancer statistics being 1 in every 3 men, 1 in every 4 women will have cancer during their lifetime. It's not that surprising that it has touched my history as well. My mom lost her dad to cancer before my parents were married. So I think I grew up without getting to know Grandpa Bill, and just always with this idea that cancer was just this incredibly sad thing. And in high school was kind of the first time I was really just struck with the idea of like, I don't want this disease to be so sad anymore. I'd like to be someone who can make a difference in this field. So I guess at that time in high school, I sort of made a resolution of like, I want to do something that's going to help other people. And then when I started school at the University of Texas for undergrad, I found this major where I could take my love of math and science and engineering and combine that all into biomedical engineering. And I was lucky to right away freshman year get involved in breast cancer research. And now that that has led me down the road to where I am today. And I'd love to be a professor someday where I can have my own lab and continue to understand this metastatic process with the hopes of stopping it.
Yeah, Katie, with breast cancer especially, it just seems like we all know tons of people who have had it. And some people, they have it and it never comes back for the rest of their 40 years or whatever. And other people, especially younger ones, it comes back. So this will be where you make a difference?
I hope so. Yeah, and I think breast cancer is a really great case study of where research has really changed outcomes. I think a while ago, you would get a diagnosis of breast cancer and that was horrible, but now it's often a very survivable disease. So I think that's where my interest in metastasis really is. It's the 9 out of every 10 cancer related deaths are due to your cancer spreading to other parts of the body. And especially for breast cancer as an example, often tumors will move to the brain or the bone.
Yeah.
So
others just stay right where they are. Did you read The Emperor of All Maladies?
I did. That was definitely
great.
a favorite book of mine.
Yeah.
I have it on my list to read again this year.
Oh yeah, it's one you need to read every 10 years or so.
Yeah.
Any of the other scholars want to comment about this? Do we have another question for the scholars, or do you have any questions for us?
I have a question for, go ahead,
Go ahead.
Jessie, go ahead.
No, I was just going to speak. Yeah.
I actually have a question for you. You're at Emory. Do you know Dr. Michael Davis, who does
That's my advisor.
That's your advisor.
Oh my gosh.
Oh my gosh, because he gave us, you know, he was showing us the laser printing of the bio heart patches. And
Mhm.
oh my
The
gosh, that's
printers.
exactly where we were.
Yeah.
Oh, I'm so excited. Well, good. That will, that's fabulous. Say hello from ARCS.
Will do.
Isn't
Yes,
that-
I didn't realize that he was an ARCS scholar himself until I received the nomination, and he told me, and I thought, what a funny connection. Yeah. So this is the 3D printing room I'm sitting in.
What
So
an exciting
how
time.
does your RNA research relate to the bio 3D printing? How does that?
It's somewhat of a separate arm. So our lab is interested in biomaterials and cell therapy. There are a few people working on the connection between the two. So I'm interested in the cells that we use. We collect patient cells from CHOA, Children's Hospital of Atlanta next door. And then I sequence them and figure out, I can predict if they're going to be good or poor cells. And then actually that information can flow directly to other grad students in the lab who then use those patient cells and apply them to their patches or their hydrogels. And then we can test those in vivo. So we're trying to work together to bridge cell therapy and biomaterials, because we know we need the best cells, but then we also know that we need the cells to stick around, because oftentimes when we use cell therapy, those cells are automatically just flushed away by your body and the immune systems that we have. So we know that if we can incorporate them in cardiac patches or other hydrogel-based materials, that they'll stick around and really have the most reparative potential. So it's kind of dual processes.
Pretty interesting research.
Very. And Christian, I was reading about your research, and one of the things that I thought was so interesting was that the mold on corn can cause early puberty in girls.
Well, yeah. Well, so so I'll kind of answer the question of how I got started and that question in one little statement. But so I went to UGA for my undergrad, and I was going to stay on and go to grad school and work under a professor here who studied how malaria can invade the placenta, because that's kind of a rare. The placenta is kind of it's a unique barrier, and it's a very protective barrier. It doesn't allow a lot of things in and out. But malaria is one of those things, and Zika was happening. So Zika was going to be another part of that, and we were going to kind of look more into that. But she ended up leaving to go to South Carolina, and my current advisor worked on the placenta, but she studied how mold contaminants can lead to disruption to female reproductive health. And so one of the ways that mold contaminants can lead to problems in female reproductive is that they mimic estrogenic substance. And actually the one that Leslie was talking about is called zearalenone, and it mimics 17-beta estradiol, which is one of the more potent forms of estrogen in our bodies. And so basically there are these studies of young women in kind of areas around the country, around the planet, where they're more likely to have a specific type of corn mold grow, and it will actually accumulate to toxic levels, and people can ingest that those toxic levels over years of their life, and that can lead to and I'm forgetting the clinical term for early puberty, but it can lead to menarche or something. I can't quite remember, but it can lead to that in some women. You know, but this is very kind of isolated in certain regions of the world. Like here in America, that would never happen. You know, we have ways to measure the amounts of these chemicals in our diet. So, you know, or I say it can never happen, but, you know, we hope that we have protections in place that will protect us from that. But yeah, so my first project in the lab was to basically learn how these estrogenic mold toxins could maybe potentially disrupt placental function. And kind of what we found was that mice given the highest diet, which is similar to like an agricultural diet, so this would be the diet that's fed to like cows and pigs and stuff, Basically, it gave the mice preeclampsia, which is a disease that affects millions of women, in which the placenta doesn't quite grow the vasculature needed to sufficiently transport nutrients and waste through the, between the mom and the pup. And so that was kind of my first big research project. But yes, in some places around the world, these toxins can lead to early puberty in women.
Well, what about an organic diet, for example, where they don't control mold on certain foods? Is it only corn too?
No, it's not only corn. It's corn, wheat, soybean. It's a lot of things. And soybean already has genistein, which is an estrogenic compound in it. So, but yeah, it's really the main is corn and wheat are the two types that this specific mold can grow on. But places that can't control the amount of mold on their product, that would obviously be dangerous. But it's actually also a big. It's not just like, so like when a farmer goes and like plows the whole field, obviously there's some mold just in that, but actually a lot of it comes from when it sits in silos. And basically it sits in silos and the mold can grow and it has the perfect conditions to grow. And then later when it's processed, none of the mold gets removed. And so then it just goes into, whatever, the city or whatever that's using. So that when I said it was very isolated, it's kind of in smaller, more rural areas where the grain that they're harvesting is just going straight to the consumer. It's not being processed into like chips that are going to the whole country. So it's kind of what Leslie was asking about is very isolated events where basically these exposures were just they weren't found. They just had no relevant means they weren't testing their corn product to see how much of these toxins are in there. They're just feeding them to people. But so what we were looking at is more at like a lower dose. Can that affect women's health? In America, because we're allowed to eat this stuff here in America. We eat it every day. So because we're allowed a certain amount in our diets just by the USDA, they say or the EPA, they say it's okay. So we were looking more at like, how does low dose over time affect women's health? But what Leslie was asked about was more of kind of like isolated events around the world where like these people were exposed to huge quantities of this stuff. And it caused these kind of population health outbreak kind of stuff. But we don't study any of that.
Well, we are out of time, unfortunately, because it has been great to visit with you scholars, and we thank you for your time this morning. And we will share a recording of this conversation with Northside. I know they'll be very interested in hearing what wonderful work you're engaged in. And we thank you for the work that you're doing to advance science. And we're very proud to have you as ARCS scholars. Thank you.